Namaste, good evening. On behalf of Central Zoo Authority and Sri Chamarendra Zoological Gardens, I extend a warm welcome to all our listener participants who have joined us today from across the globe. This is a third in series of the webinars that we have initiated to reach out to the zoo community on topics that are of mutual interest and also to bring to you global experts who are well known and respected in this field. Today, through this digital platform, we bring to you some very esteemed speakers to discuss and dwell upon the aspects of biobanking, cutting-edge science to reduce extinction risk. At the onset, I would like to welcome Dr. S.P. Yadav, ADG Project Tiger and Member Secretary, National Tiger Conservation Authority and Central Zoo Authority. A keen forester and wildlifer, Dr. Yadav has been instrumental in steering tiger conservation in its range countries and is always supportive to out-of-box approaches and capacity-building initiatives to strengthen ex situ, in situ linkages. Over to you now, sir, for your opening remarks. So we cannot hear you, sir.
Am I audible now?
Yes, you are.
Yes. Am I? Yes. Yes. Okay. Okay. Thank you, Sonali, and good evening to you all. And on the behalf of CZA, I warmly welcome Dr. Ryder and Dr. Karthikeyan, and wish that this webinar will be exciting to all participants. Dr. Ryder, very good morning to you because it must be early over there. And
thank you.
Dr. Ryder, if you recall, I was privileged to attend a session on biobanking in the last WASA conference at Buenos Aires. So that was really nice and quite insightful, quite informative. So basically, the Central Zoo Authority of India is a statutory body and works under the Ministry of Environment and Forest and Climate Change. And this is statutory in the sense that its existence has come because of the Wildlife Protection Act. Unlike other countries, the CZA has powers to regulate the establishment of zoo. No zoo can be created or can operate without the recognition by the CZA, by getting recognition from the Central Zoo Authority. So in that way, we do regulatory function. Gradually, we are graduating into facilitating role. We also provide technical assistance and financial support to Indian zoos. And you may be aware that there are 152 recognized zoos in the country. And Dr. Ryder, you will be surprised to know that there are more than 70 million people that visit our zoos on an annual basis. Zoos are very good centers for education and awareness, especially for the kids and the aged people, because they cannot visit our national parks, sanctuaries, and tiger reserves. Next slide, please. Why this biobanking is important, I think everyone understands. I recall a few months back, United Nations IPCC report that was alarming, kind of. It said that 1 million floral and faunal species, they are under threat of extinction. And the rate of extinction is much faster than what we have experienced earlier. So definitely the situation is very grim. If you take example of India, we lost our cheetahs in 1947. Last cheetah was sighted in 1947. And after that, it's more than 70 years, we have not been able to bring back cheetah into our wild. Similarly, if you see the example of Tasmanian tiger of Australia, lost forever, it is obvious that once we lose the species, we lose it almost forever. There are a number of species in India which are at the verge of extinction, like brow-antlered deer, the great Indian bustard, that bird. Hardly 150 specimens are in the wild. Similarly, Kashmiri stag, there are a number of species. So the role of Central Zoo Authority is to support the in situ conservation by doing conservation works in captivity. So I believe that this collection of genetic material from dead specimens is the most easiest thing which can be done to collect the genetic material from the animals, wild animals from our zoos. We have LaCONES. I think the details will be told by Dr. Karthikeyan. He represents LaCONES, and they are doing a wonderful job. So CZD has prioritized number of species depending upon their status of endangered. Next slide, please. I believe there is lack of awareness about biobanking. There is lack of capacity and lack of knowledge about the data management, like what to do after that. I also believe that for biobanking, a regional and global network is very, very much important. And knowledge sharing, above all, is definitely very important for all of us. And for that purpose, this webinar has been organized. Dr. Ryder, I feel that down the line, 20 to 25 years, we'll not be exchanging animals, but we will be exchanging genetic material, and maybe we recreate the animals. And that is, I think that time will come, which will, with this available technology, it will enable that. So we need to create a consortium of zoos to participate in the advances of biobanking for wildlife genetic resources. Definitely this technology, this facility, that this needs funding resources, infrastructure resources by the government, and creation of capacity, that is also very, very much important. And I think with this webinar, our zoo participating zoo directors, they will understand the importance of biobanking, and we will march ahead in this direction, because I see a great future in biobanking and cryopreservation in supporting in situ conservation in the country and the world. Thank you very much. I'm excited to hear Dr. Karthikeyan and Dr. Ryder. Thank you so much.
Thank you very much, sir. You've set the tone for the webinar today. So now I would like to welcome Dr. Oliver Ryder, Director of Conservation Genetics for San Diego Zoo Global and Adjunct Professor in the Department of Evolution, Behavior and Ecology in the Division of Biology at the University of California, San Diego. Dr. Ryder directs a laboratory group that includes activities in the area of molecular genetics, cytogenetics, cell culture, tissue culture, and stem cell studies. He has been involved with San Diego Zoo's Frozen Zoo project for over 35 years, and this unique resource of cell cultures has contributed to genetic rescue of several critically endangered species, including notable scientific contributions to advancement in technology. Over to you now, sir, as this must be very early morning for you. So thank you again for joining.
Thank you. I'll share my screen when I can. And I'm honored to be invited to participate in this important discussion. I will share my screen. And we can get started. So I very much appreciate the invitation and the comments from Dr. Yadav. This is, I agree very much. This is a very important topic, and it's one of global importance, and it's one that benefits both the current efforts that we are taking now and will have a significant impact in the future. I'd like to acknowledge my colleagues who are listed here. I'm privileged to speak on their behalf. And we have a modest effort in tissue culture and cytogenetics and cryobanking and also gamete preservation at the San Diego Zoo. And the San Diego Zoo operates 2 facilities. I'll say 3 here. They operate the San Diego Zoo. They operate the San Diego Safari Park, which is 80 kilometers north of San Diego. And San Diego is in the very southwest corner of the United States. We are 75 kilometers from the international border with Mexico. This research facility is called the Beckman Center for Conservation Research, and it's where the home of the frozen zoo is, whose picture I just showed you in the previous slide. The frozen zoo was started in 1975 by the founding research director for the San Diego Zoo, a physician named Kurt Benirschke. In 1986, I had the privilege to take over that work on the frozen zoo, and was aided initially by Arlene Kumamoto, and now the frozen zoo is curated by Marlis Hauk, who's become an international expert, recognized expert on the banking of cells. The frozen zoo has contributed enormously to a variety of studies. So as it was initiated, we didn't really appreciate all of its potential uses, although we did recognize that it was going to provide a resource that would enable studies and applications that we couldn't imagine at the time. But key papers and significant contributions in the area in these areas that are listed here, in phylogeny, in systematics, and in species definition, and in the very exciting, expanding area of genetics in the last 5 decades. And this information, having these materials banked, has allowed significant studies that have contributed to population management. And now, with recent events, recent discoveries, it is possible to use these frozen cells for gene flow into living populations. And for the first time in the history of life on Earth, it's possible to have living genetic material from an animal that's died or a species that's gone extinct. This provides, if its potential can be realized, an extraordinary new paradigm for the conservation of biological diversity. We don't have all the technology to apply to do all of this right now. We do have it for some species. It's actually possible. So there's a proof of principle. But in the future, as technologies develop, and as we would anticipate that it would be possible to undertake more and more cellular interventions to preserve species using cryobanked material, the key feature will be, did anybody bank the material? And there's never going to be more biodiversity than there is now, at least not for millions of years. So we are at a very critical point in time where technology is being enabled, where biodiversity is declining, but that we can be agents of transferring the potential for an unprecedented level of species conservation to the future. And just to keep things current, these frozen zoo materials are providing an extraordinary and powerful and sought after resource for expanding our knowledge of comparative genomics and biodiversity genomics, and genomics that are applied to the current pandemic to understand the evolution of the receptors and viruses that are causing that are contributing to the current pandemic. I would say too that having living cells allows you to expand your knowledge of chromosomes. You can only visualize chromosomes in dividing cells. And so we, in our process of accumulating samples, just randomly, we received a sample from a tiger in a zoo in the United States, and it turned out it has a chromosomal error. This chromosomal error is similar to a human syndrome called Klinefelter syndrome that leads to sterility. And in fact, this male had been fruitless in efforts to reproduce for a number of years. And then we knew why, and steps could be taken. But there are this is just a quick example of some of the health interventions that can come from having these cells. We found a gorilla that was growth retarded. And when we studied his chromosomes, we found out he had a big deletion in his third chromosome. So this individual is excluded from the breeding population because of this genetic error. And it helps us in understanding and managing the life in captivity of this particular male. The frozen zoo has accumulated samples, I said, since 1975, and it's been a steady and ongoing enterprise. Year by year, more species are added, more individuals are added, so that we've accumulated a large bank. So when people are starting out, it's important to remember that the history of this kind of enterprise is very much making a modest start and building it up, but it's important to get started. My colleague, Dr. Barbara Durant, banks reproductive cells, ova, spermatozoa, and reproductive tissues. So the frozen zoo includes both these resources. I'm going to talk mostly about the fibroblasts, the cellular resources that we can divide and expand. But just quickly to go over what the collection looks like now. In cryobanked sperm, there are over 400 species and probably about 2,000 individuals now, since this is an older slide. And there is the breakdown of their taxonomic classification. Most of them are mammals. The same thing is true for the fibroblast collection, the skin cell collection of the frozen zoo. It's mostly mammals, and of the mammals, it's mostly primates, perissodactyls, artiodactyls, and carnivores. But the fastest growing portion of the frozen zoo is birds. In the last few years, more new bird species have been added to the frozen zoo than mammal species. As a result of our work over the years, we've now accumulated many mammal species. So this slide is a bit old, but here's a general breakdown of the numbers. And it's got very broad representation across terrestrial vertebrates. And as I said earlier, this has facilitated a large number of studies. I think that the scientific community is largely unaware of the crucial or very important role that collections like this that come from zoos have contributed to the basic understanding and of biology and basic scientific knowledge. We started a program to try to save cell cultures from endangered amphibians. It's estimated, if I remember correctly, approximately 1/3 of amphibian species are in the threatened classifications in threatened, endangered, critically endangered. Many of them are threatened by diseases, chytrid fungus or viral diseases. And when we want to manage and learn how to, manage and treat and prevent diseases, we often rely on in vitro systems to study this so that the studies don't have to take place in live animals. And this is especially crucial for endangered species. but there was and still is a significant underrepresentation of these critically endangered species in cell banks and in the ability actually to even culture their cells. So we've had to undertake really very basic studies to learn how to culture amphibian cells. And while we're very proud that we probably have the largest collection in the world of diversity in amphibian cell cultures, it still fails in our hands most of the time, and we need to do basic research to learn why. So just to kind of summarize or reiterate, there are many important reasons for banking living cells. They're expandable. If you freeze them at early passage, you have them stored for millennia. They can be taken out and additional cells can be grown. You can freeze back and add back to the bank more cells than you took out. It's not an infinitely expandable resource because these kinds of cells senesce in culture, but you can manage this and curate this to be able to produce a very large amount of material that can be kept for the long-term future. And we should be thinking about this, not just in years or decades, but on a very much larger time scale about how the future can benefit from having this kind of material. These kind of living cells is an exquisite source of DNA from any organism, but from many small organisms, using current technology, it's the best and perhaps only way to obtain sufficient amounts of DNA for certain studies. And this includes enabling the highest quality genome assemblies. This work in comparative genomics and in conservation genomics is adding enormously to the field of linking ex situ and in situ conservation. Excuse me. And the having cellular material has made the frozen zoo a preferred resource for some of the cutting edge comparative genomic studies that are taking place now, including, well, I'll mention that in a minute. And with cells, you can do tests, as I said, in vitro that don't require the use of animals. This is very important for understanding basic biology and how cells work and how organisms work and how diseases progress. And remarkably, these cells can be used to produce induced pluripotent stem cells, cells that can make any cell in the body. And that means that the potential for banking these skin cells is to actually produce animals, either through stem cell technology or through somatic cell nuclear transfer cloning. This has been done in model species, and if this could be done across the species that are threatened and endangered, and it would, as I said earlier, dramatically insert a new paradigm into conservation. And when you work with cells, you obviate the necessity to handle animals, so it reduces animal welfare concerns. And I think unequivocally, banking cells serves the interests of the future. So this field of genomics now, over a decade ago, a project to sequence 10,000 vertebrates, which was audacious at the time, called Genome 10K, began. It's now morphed into a project called the Vertebrate Genomes Project, which really aims to sequence the genomes of all vertebrate species. The technologies have improved, the costs have decreased, there are global centers, there are now DNA sequencing instruments that can be taken into the field and run off laptops. This is a very rapidly and excitingly developing area of biology that has direct relevance to conservation of biological diversity by assessing and monitoring biodiversity, characterizing it, and using that information to manage gene pools for long-term sustainability. In Nature, later this year, will appear a paper about the 200 Mammals Project that was a collaborative study with many institutions, but led out of the Broad Institute by Eleanor Carlson. And it's produced some remarkable findings, both that are relevant to human medicine and to endangered species conservation. I don't have time to go into all of this, but for those of you interested in promoting or evaluating the importance of biobanking and cryobanking cells, this paper will be very useful for you to cite. So one of the real potential applications is for what we can call genetic rescue. Genetic rescue is a proven method for reviving species that have become populations that have become fragmented and are declining by infusing new genetic variation into these dwindling populations. Notable examples include a large cat in North America called the Florida panther, which animals were used from an adjacent population, although they were considered a different subspecies, and a transient gene flow into the Florida panther population led to its invigoration and the reduction of the problems associated with inbreeding. And this has now been noted in avian and other mammalian species. And so we know that it works by transferring animals, and this could in the future potentially be accomplished by using advanced cellular and reproductive technologies. And the inset there is a picture of a cloned Javan bantang that came from cells that had been stored in the frozen zoo for 25 years. And this animal had genetic representation that was gone from the ex situ population. But by cloning this animal, the opportunity to introduce this genetic variation back into the ex situ population was manifested. As I mentioned, the possibility to produce induced pluripotent stem cells for endangered species has been identified. The first paper on this came out, I think, in 2013, in a collaboration we did with the Scripps Research Institute. And we chose 2 endangered species. One was a primate, the drill, the largest and one of the most endangered old world monkeys, and the other was a rhinoceros species. And before we get too far into the application of this technology, I want to be sure that we all understand, and the context from which I'm speaking is that the real way to preserve species is to save them in functional ecosystems and in functional habitats. And efforts need to be increased and sustained across the globe to do this. But the very fact that we have so many endangered species and that number is so rapidly increasing suggests that this strategy alone is going to be insufficient for passing on the legacy of biodiversity that current generations have, and that other options ought to be looked into, and that can make a crucial difference for what generations of humankind in the future have as biodiversity in their world. Just quickly, San Diego Zoo also has a plant conservation division, and our cryobanking and effort includes plant conservation. Most of our plant conservation work is done with a seed bank, but we have many endangered species of plants in San Diego County. And so our focus right now is local on species, some of which are very difficult to preserve as seeds, like oaks, which produce acorns. So we're exploring cryogenic methods of preserving germplasm of a number of endangered species. So we think that a really global approach is needed to bank all kinds of living material. And as Dr. Yadav said, this really needs to be looked at through the development of regional and national-based efforts to cryobank. just quickly, these are all species that we have participated in banking cells from the last living individual. The process of extinction is not abstract for us. It is for many people, I think. An animal disappeared and they don't know, but we have had living tissue material. We have processed it. We have helped others to bank the last individual of a species. And it's for two of them here, it's successful. For the gastropod, for the snail, and for the frog, we're able to bank living material, but we don't know how to grow the cells yet. We know how to preserve cells cryogenically, but we don't know how to actually propagate the cells of some of these species. So this leads us to a sense of, and saving one individual isn't going to save a species. The need to intervene comes earlier. These are species that we're working on now that are literally on the brink of extinction, and that we are trying to expand our efforts to bank cells for these species, because we're confident that they'll be useful in the future. So I want to just try to impress that this methods of cryobanking living cells can really make this time be one that that adds to successes in reducing losses of biodiversity. And we need hopeful stories like this. And for some species, this may be the only way to save them. And the ultimate success of an effort like this really depends on how much has been banked. And nobody's going to be, and we're in a position to do that now. And the need for expanding this capacity is an urgent concern for our time. Quickly, the frozen zoo of all of its successions, Andrew Mooney, a PhD candidate and Fulbright fellow, examined the collections of the frozen zoo. And here the numbers show for the number of species that we have in these various categories. And you can see that we have a substantial number of species in what I would just now call the threatened categories. We actually have 5% of all threatened birds, amphibians, and reptiles, at least some individuals represented in the frozen zoo, which is an enormous accomplishment. But it means that 20 times more are not represented. And we don't know, they may be represented someplace else in the world. We may not have access to them. We have 50% of all of the extinct in the wild species. But if we were to, just looking at the inventory of zoos that is kept in ZIMS, the Zoological Information Management System of Species 360, if all of those zoos, which are going to have those animals, which can access them even after they've died, we could triple the number of taxa that are cryopreserved. So this is an important, so zoos play a very important role. And this has been recognized through WAZA, the World Association of Zoos and Aquariums, at a recent meeting of WAZA in Bangkok, to pass a resolution to recognize the importance of this activity and to encourage WAZA members to participate in an accelerated plan to bank viable tissue culture cells, and to create a global database so that we know who has what, so we know what the priorities are, so we can help develop a plan for identifying priorities. Just very quickly, I need to close, but I want to just give one example that is a very ambitious one That is to try to use these technologies to preserve a species that's on the brink of extinction, the northern white rhinoceros. And I said species, and I really meant to say unique form of rhinoceros. The northern white rhino is only 2 living individuals now, both females. But we have banked cells in the frozen zoo over the last 40 years and have the cells of 12 individuals. And we've sequenced the genomes of these. The southern white rhinoceros, its closest relative, went through a genetic bottleneck a century ago, down to a low of between 30 to 100 animals. So we sequenced a similar number of southern white rhinos and northern white rhinos, and in 12 cells, so the cells of 12 individuals in the frozen zoo, there's more genetic diversity than there is in the entire living population of southern white rhinos that are approximately 16,000 individuals. And we can see the signs of the inbreeding that took place after the genetic bottleneck in the southern white rhino, because they're larger stretches of a genetic identity that come about when individuals have relative the same individual on both sides of their pedigree, from the maternal and paternal sides, when they're inbred, in other words. So we can detect these things now and actually study genetic load. And we've made fibroblasts from those 12 individuals. We've made induced pluripotent stem cells from 9 of them. And we're able to direct the development of those. These are beating cardiomyocytes, beating heart tissue, spontaneously beating in the laboratory to show that these cells are indeed pluripotent. We hope to be able to make gametes, and that's a sort of an extended story. I'd like to acknowledge all of the people in the lab that have contributed to this, or at least the current lab members, and thank you for your attention.
Thank you very much, sir, for an excellent presentation. I'm sure there was a lot of things which have been an eye-opener for us, new things for us, and also give us hope. I must admit that while researching for your talk, I fondly read your blog about Nola and Angalifu, the northern white rhinoceros at San Diego Zoo, and how they continue to be a beacon of hope. So we request you to kindly stay on, and if time permits, we will request you to respond to a few questions. So now I would like to request Dr. Karthikeyan Vasudevan, Senior Principal Scientist at the Laboratory for the Conservation of Endangered Species, LaCONES, at Center for Cellular and Molecular Biology, Hyderabad. He has also been appointed the recent expert member for the Central Zoo Authority. He and his team at the National Wildlife Genetic Resource Bank of LaCONES CCMB have developed a repository for germplasm and genetic resources of wild animals since its formative years, and have developed techniques to cryopreserve for around 23 species. His present work is to use biological research and mobilizing technologies that will strengthen conservation of endangered species in this country. Over to you, sir. Dr. Karthik, we cannot hear you.
Can you hear me now?
Yes.
So I was just saying that Oliver's talk was really fantastic, and we could, I could make my it has eased out my job, because it has given a fantastic overview of what are the possibilities and the rigor with which the San Diego Zoo is developing the biobank, and how useful it has been for several species, and also understanding of biology. So my effort here is to bring forth to the audience of this webinar of the effort that CCMB has been taking up to create a repository for endangered species in India. And obviously the source of all the samples would be from, majority of them would be from the zoos. And the whole concept of the laboratory for the conservation of endangered species, since its inception, one of the most important aspects has been to develop this biobank. And since 2007, when we had this institution, the physical infrastructure being put, we have grown in the collection over the years. And I'm going to take through the journey of the collection growing in this lab. So, so we all know that the assisted reproduction technologies are very important for rescuing endangered species, and Dr. Oliver has already given us an overview of the possibilities that present when we do biobanking. And usually when a species comes to the point where it has to be rescued, uh, it is in a very small population. We call it a founder population. And if the founder population is highly inbred, or it is not representative of a diverse genetic constitution of the original population, then very quickly the number of individuals can reduce, or infertility can set in, and the species can go extinct. So the reproductive technologies have been very well streamlined for domestic species. We know we use them for increased production of dairy products, poultry products, and all of these technologies can very suitably adapted to address requirements of endangered species. And I've just listed a few of them, and there are more. And the reason I list them is because in all these aspects, LaCONES has been contributing to be able to implement assisted reproduction technologies in endangered species conservation. So it starts with reproductive monitoring. You can use hormone assays, fecal hormones, or residues of hormones in sweat or saliva. They can inform about the reproductive condition of the animals so that interventions can be taken up. Artificial insemination is a well-established technique, and however, it requires a lot of standardization, which I will just mention in my next few slides. And then how reproduction can be controlled, how can we induce estrus, cause superovulation, and be able to harvest oocytes from the female. All those techniques involve repeated experimentation and standardization. And then finally, we have the capability to produce embryos in vitro, preserve them, and use them in surrogate females so that we can give birth to animals of our endangered species which are required. Then, as Oliver Ryder pointed out, they are important genetic resource banks, and they generate a huge amount of information for people who would like to explore the diversity of life and understand evolution, and even identify very important species phylogenetically. So I'm going to rely upon the work that has been done by two of my colleagues to take you through this talk. One is Dr. Samba Shivarao. He's a female reproduction biologist, basically looks at the female side of reproduction. And Sadanand Sonteke has been, both are veterinarians, and they have trained to handle animals. And Sadanand has expertise in euthanasia, and, anesthesia, and male reproduction. And the whole problem of working with endangered species is that we have to repurpose many of these technologies to address all the wild animals, and each of them have distinct reproductive anatomies, and their requirements for restraint are different, and collection of these biological materials is very tough. And for all of this, the cooperation with the zoos are very vital. And this is exactly what I wish to highlight in this talk. And these are images of the ovaries of different animals, and even people who repeatedly perform postmortems will find it difficult to identify these small structures in the body, and it requires a well-trained eye to pick up these things and be able to harvest them and bring them into the biobank. So the main aspect of biobanking is how we can bring endangered species genetic material into the biobank, and you have mortalities happening periodically in the zoos, and when these animals die, if we can bring these animals to conditions where their tissues do not degrade very fast, we can harvest testes, ovaries from these animals and cryopreserve them and be able to use them for future cluster reproduction technologies. So there are several examples of this that have happened in our lab, and we have a variety of tissues that we collect from both postmortem and live animals, and this is just a list of those samples that we collect, and this bank is slowly growing, and way back in 2007 we have been listed as one of the centers that store genetic resources on wild animals, and we had the talk from Oliver mentioned about the frozen zoo in San Diego, and the frozen zoo is the motivation behind having the biobank in LaCONES. And we had Samba Shiva Rao is here. He went to San Diego Zoo to get trained in 2016. He attended a workshop there, and he is now managing this facility, which is called the National Wildlife Genetic Resource Bank, which was dedicated to the nation in 2018. So we have facilities which we would like to almost mirror what the frozen zoo in San Diego does, and be able to grow like the way they did after we obtain the required cooperation from all the zoos in the country. So the steps that are involved in biobanking genetic resources, particularly for the females, is that we have to find the ovaries in the females and be able to bring them to lab, and then recover the oocytes, and then mature them, and then cryopreserve them, and then finally we can use them for fertilization using IVF or ICSI. So all of these requires timely access to the samples. So we have been able to perform biobanking of oocytes of several herbivores. This is just an example of the variety of herbivores that are being cryopreserved in our repository. And you can see that there is a drop-off from the number of oocytes recovered to the culture grade oocytes and then mature oocytes. This is an artifact of the whole work, because every time there is a loss of some viable cells, and then and the whole effort is to maximize the potential of all the cells that we collect from the dead animal. So we are trying to make it better for several species, and for all of those who don't know the process of cryopreservation, for each of these species, the effort has to be standardized independently, and they can vary from one species to another. So it involves some tedious experimentation. So the same has been done with several felids. You have tiger, lion, leopard, all of them we have collected oocytes and been able to preserve them. And then other mammals are also there in the collection based on the access to the samples. We have been able to collect these species. However, if we expand our collection, we might be able to cover more species, and which are certainly important to be covered in the biobank. So with the male gamete, we require sperms. The testicular tissue itself is an important source. So when we have post-mortem collection of these organs, we look for harvestable cells and be able to preserve the semen from the chorda, or be able to, if we have live animal, electroejaculate it and collect the semen and cryopreserve it. So all of these has resulted in identification of potent males, because zoos are interested in identifying males. Like in the graph on the left, you can see the black buck, which have large number of sperms in the ejaculate, are certainly candidate males to be used for reproduction. So the studies on the male reproductive system has led to improved breeding success in the zoo conditions. And you can assess sperm motility, and then be able to cryopreserve them and use them in subsequent artificial insemination efforts. So we have had success with artificial insemination in ungulates. In the case of black buck, we have been able to produce live offsprings twice, and we have had several insemination attempts. And this, at least for ungulates, we can say that the methods have been fairly standardized. So another repository of skin tissues, fibroblasts, skin tissue culture that we maintain, what Oliver mentioned, is also being done here. And we have several species of ungulates, felids, canids, primates, and reptiles. Or these numbers don't match with what the San Diego Zoo has already done. But we hope to grow in this area, because every time when we get a viable tissue, then we start work on them and try to maintain cell lines of those tissue sources. So I'll just give you an example here. Just because we could collect several viable tissues from big cats, particularly tiger, we are in a position to also help today with identification of tigers which are seized in the forensic cases. So we are standardizing the markers, and maybe in future these cultures will help us produce a DNA ladder of all the microsatellite markers, so that it can be used as standard throughout the country to be able to identify individual tigers which are seized in for forensic examination. So the spin-offs of such repository are completely unprecedented. We cannot even imagine what the use or application might come to be of value in future. So we have to do this exercise repeatedly and carefully to be able to increase the collections in our biobank. So this is just a representation, not updated to the current year, but a large number of animals are in our zoos in India. So obviously animals die, and when they die, there is a post-mortem and a report is made, and then new animals are sought to improve the collection. So it is just a process of bookkeeping, which is maintained by the zoos. But till the time after the death of the animal, there is no use. There is no the reproductive potential of the genetic resource of that animal is not available for future. So the idea is be able to perform in a better way with these resources and achieve fantastic outcomes like what has been done with the black-footed ferrets, or the northern white rhino, or the cheetah, which has been recently produced. Live birds have been produced using in vitro fertilization of sperms that were stored in frozen condition. So all of these big outcomes may not come about if we do not do the biobanking like the way the San Diego Zoo has been able to do. So the proposal is that we create a consortium of Indian zoos to begin with. Zoos can join in future, but initially it will start with a group of zoos that have a large collection and be able to communicate with them in a coordinated way to be able to access the samples as soon as there is a death reported, and these samples are brought to the biobanking facility, and there is also a two-way communication between the biobank and the zoos, and be able to create capacity within the zoos to identify the organs of importance and collect them, preserve them in proper manner so that they are viable for the biobank. And as I told you, the interaction has to be two-way, and the biobank has to facilitate zoos and tell them that the animals that have died are in fact live in some form in the biobank, and that can also be a form of communication to the visitors of the zoo to inform how science is helping in the zoo management and conservation of endangered species. So I would leave this with a quote from Kurt Bernishka, who's the founder of the Frozen Zoo. I think Oliver mentioned about him, and because of his ideals, we have the San Diego Zoo presently functioning the way it is. So he said, you must collect things for reasons you don't yet understand. And I think that's a very strong statement, because we don't really know what would be the utility of these things. So we have to do it, because in future they might come of the important use. Thank you.
Thank you, sir. That was excellent and fantastic in terms of the kind of advances we have in our country. And also we heard Dr. Ryder for the global context, and we are doing wonderfully on time. So I think I will pick up 3 questions for each of our speakers, and I'll request Dr. Ryder and Dr. Yadav if they can be available. Dr. Ryder, sir, there has been a very interesting question in the chat box of the YouTube, and I'm going to ask you that. Is that what basic facility should a zoo need to contribute to the Frozen Zoo initiative or to the cryopreservation facilities?
Thank you very much. And let's begin by commending Dr. Karthikeyan Vasudevan. I'm sorry if I didn't pronounce it right, but that was a very good talk. And I'm really pleased to learn about the activities that are taking place in India, and commend the organizers of this webinar series for really helping facilitate international exchange of information, especially in this time. But to answer the question, having access to animals is the key feature. Collecting a skin biopsy is not difficult. It can be done by people with very basic kinds of training. And so I think the requirements to participate are minimal. For an institution that doesn't have a research laboratory that's going to process the sample, they need to coordinate closely with the entity, the research lab to which they're going to send the sample, so that the right media can be used, the right procedures can be in place. But There are now, if they're not available already, they will be soon. Things like YouTube videos that explain how to do this. So for people in zoos or field researchers. So the important thing is to recognize that individuals that have access to an animal can provide can play a very important role, and it's not that difficult. Thank you.
Thank you so much, sir. The next question I would like to give to Dr. Karthikeyan Vasudevan. Sir, what stage is the project Asian cheetah, and is cloning being actively considered for its revival in India?
So, am I audible?
Yes.
So, yeah, this has been repeatedly asked, and in fact, LaCONES had it as one of its main objectives when it started way back in 2001. But if you recollect the sequence of events that happened, we had an agreement with the government of Iran, and they had to spare a few cheetahs from Iran to be able to. That was identified as the closest, cheetah that would have occurred in India. So, but the treaty failed, and we didn't get the cheetahs from Iran. So we had leopards, and we worked extensively on intrauterine insemination in big cats, and we tried several artificial insemination techniques with big cats. And people who know the reproductive biology of big cats will understand how difficult it is to perform any assisted reproduction technology with big cats. So, to give a very simple answer, the project has presently been abandoned because we don't have the genetic material for the cheetah, Indian cheetah, as well as the closest relative of the Indian cheetah.
Right, sir. Thank you. Yes, I would like to now ask a question to Dr. S.P. Yadav. So the question has come that India has done relatively well in situ conservation. So is there a need to invest, if it is in terms of funds, into the ex situ technological options, such as the biobanking and cryopreservation?
Okay, that's true that India has done excellent in situ wildlife conservation. Like, we are the largest tiger-rich country in the world. We have more than 75% wild tigers in our country. We are largest single-horned rhinoceros country in the world. We have largest Asiatic elephant population in the world. We are only home to the Asiatic lion in our country. So success is there. But as you have you have heard and seen both the presentation of Dr. Ryder and Dr. Vasudevan, extinction is not abstraction. Extinction is happening every day, and it's very world over. We have seen example of northern white rhinoceros. We have heard example of Indian cheetah. So I think the only way, and there is in Chhattisgarh, the state animal wild buffalo, there is no female available other than the one cloned one. So this kind of things are happening every day. We have to be ready for the future. I think this all makes the investment in cryopreservation or biobanking very justifiable. Thank you.
Thank you very much, sir. I think we will close here because we are actually running short of time now. And I would like to end this webinar with a big thank you to all our esteemed speakers. Sirs, we would like to continue this collaboration, and we rest assured to everybody that the Indian zoos and specifically Central Zoo Authority will forward this first capacity building and strengthening that we did today and into some fruitful collaboration. I thank both the teams at Team Mysuru and Central Zoo Authority for helping out in the smooth functioning of this webinar. And the recording would be available, and we will also share the speaker presentations with everyone. Thank you so much.
Thank you. Thank you very much.
Thank you, Dr. Ryder. Hope to see you soon. Maybe because this time this WASA conference is not happening. Otherwise, we would have physically met you at San Diego. But let us see what happens next year.
I'm sorry that we won't have the opportunity to meet in San Diego, but let's hope for the best.
Yeah, right. Thank you. Thank you, Dr. Vasudevan. See you sometime.
Thank you. Thank you, Oliver.
Great talk.
Thank you. Great talk from your side as well.
Thank you.
I'll tell Sambashiva that I met you.
That's very good. I'm so pleased you showed that picture of the frozen zoo. So